Hi, everybody. We're back to part six, the historical foundations of microbiology from chapter one, the main themes of microbiology. Now, since I'm doing each of these individual videos as sections, chapter one has a lot of these sections. There are seven. Most of your chapters will only have three or four sections, so there won't be as many videos to watch. But I thought that this would make it a good way for you to just be able to target the areas. If you wanted to go back and just look at one part of a video or you didn't understand something, you could easily just go back to that one particular video instead of having to screen through a 3 hour long video. Anyway, let's get to it. So the learning outcomes here are to make a timeline of the development and history of microbiology, list some recent discoveries, and explain why about the scientific methods. We're going to kind of learn a little bit of review stuff. So some early ideas about disease transmission is people understood that foods could become spoiled or become inedible, and that they would sometimes cause illness, like diarrheal diseases or vomiting, things like that. But they didn't really understand what was happening in the food that was causing that. We now know that it's usually things like bacteria or other things, which is why we keep our food in the refrigerator or freeze it or cook it really well and eat it right away and things like that. um, Black Plague and the smallpox, they understood that those things were caused by some kind of transmissible thing. They understood that it was being spread from person to person somehow, but they didn't understand what those things were. We now understand them as bacteria and as viruses and have a better grasp on really what the agents are themselves that are causing the disease. And people had this belief in spontaneous generation. So what does that mean? That really kind of means that they thought that if you left a piece of meat sitting on a countertop and later you saw maggots on it, that those magnets spontaneously appeared. And just magically appeared. We now know that that's not where maggots come from. Maggots come from flies. They're fly larvae. So I have this video I want to show you, and it's a little bit long, so it's going to add to the length of this particular PowerPoint. But I think it's a good video, and it actually will cover a lot of some of the other topics throughout. So it'll make it faster for us to go through the other things. So let's watch this now.
Long ago, it was believed that living creatures originate spontaneously from the non-living matter. Although there was no scientific theory supporting spontaneous generation, people have developed this concept by their routine observations that occur around them. Aristotle was the first one to record his observations on spontaneous generation. He described about the formation of maggots from decaying meat and sudden appearance of rats from stored grains, etc. However, his concepts were remained unchallenged for more than 2,000 years. In 1668, Francesco Redi has first disproved the theory of spontaneous generation. His experiment has demonstrated that maggots arise from flies, not from the decaying matter. Later, there were many experiments and arguments on spontaneous generation. In 1676, after the discovery of microorganisms by Leeuwenhoek, the arguments on spontaneous generation have taken a new turn. Many scientists have proposed theories that supported spontaneous generation for microorganisms, but not for the larger living animals. In 1748, John Needham had observed microorganisms in heated meat broth that were not present at the beginning of the experiment. He concluded that the bacteria has originated from the meat. At the same time, Lazzaro Spallanzani has further improved Needham's experiment. He boiled the meat broth and completely sealed the flask, and the microorganisms did not grow. He concluded air has carried the germs to the broth in Needham's experiment. Theodor Schwann passed the air into a flask containing broth through a red-hot tube. The broth in the flask has shown no growth. Simultaneously, in 1850, George Friedrich Schroeder and Theodor von Dusch have allowed air into the flask by passing it through a sterile cotton plug. No growth was observed. They said, the cotton fibers have trapped the microbes from the external environment. The usage of cotton plugs to avoid microbial contamination exists even today. Later, in 1861, Louis Pasteur has completely disproved the spontaneous generation concept by his swan neck experiment. Let us go through these experiments.
So I'm just going to pause it here for a moment to say, pay attention to this video. You can pause it, stop it, slow it down. Make sure you understand the intricacies of what they're talking about here. So the first experiment they're going to talk about is Francesco Redi, and then they're going to go through some of these other experiments that ultimately disproved spontaneous generation. We no longer believe that. We understand that life comes from other living things. Bacteria don't just magically appear, they don't come from the meat, they come from themselves, and they, if they're present in the meat, then they can divide and grow, or they can be present in other places. So let's go ahead and watch the rest of this video. So I like this video because it does show us, it walks us through those two classic, very important experiments that disproved spontaneous generation and really helped us gain an understanding about how living things work that we take for granted today.
Long
So So this is just, we just saw the video, so I'm not really going to say anything more about it, but this is just again showing you how Pasteur's experiment worked and the fact that the bacteria got trapped in the swan neck, but when you remove that, then the air could introduce the microorganisms. Okay, so some other historical people. We have Robert Hooke. He made the first observation of microbes in the 1600s. He actually taught the first microbiology class and wrote the textbook called Microbiologica. So if you hate microbiology, you can thank him. No, I'm just kidding. But so he was around in the 1600s, so quite some time ago, and did these very detailed drawings of the microscope. He made, uh, he used the microscope in that way. Along the same time was Antony von Leeuwenhoek. This is a picture of von Leeuwenhoek's microscope. So I always want students to take a quick look at this and to think about what it would be like to use this. Here's the focusing screw. This is where you would put the specimen, and there's the lens, and so you kind of hold this up and look at it through the little lens there. When you're feeling frustrated in the lab using our new modern microscopes, um, appreciate that we have such better technology compared to Antony von Leeuwenhoek, what he had. Um, but despite that, he was able to he looked at threads, he looked at teeth scrapings, he looked at all kinds of things. He was he sold fabrics. Is his profession. And so he would ask the ladies that would come in to buy the fabrics if he could scrape their teeth. And he discovered and described, animalcules, the things that he could see using his microscope. So he was observing, protozoa and other living things through his microscope. And he wasn't heat fixing them or killing them or making slides in the same way that we were, but yet he was still able to make these very intricate drawings and details and understanding of this microscopic world that we had known nothing about before that. In modern microscopes, thank goodness, because his microscope, I don't know how many of us would be in this field. We use better lenses, we have finer focusing devices, we have light sources, all these different things that make doing microbiology a lot easier, because we can actually observe things in a much easier way and in much clearer detail and better detail. So in the 1980s, PCR was invented. The person who invented PCR, his name is Kary Mullis. He is kind of an interesting guy. He apparently did a little bit of drugs, but he described having this idea and it actually worked. And we think PCR is kind of the basis. We use it all the time in laboratories. I mean, you won't be using it in our micro lab because we do cellular microbiology, but people use the PCR stands for polymerase chain reaction. And you can really think of it as a Xerox machine or a copy machine for DNA. So to copy little bits and pieces of DNA. And it's why we can do the Human Genome Project and why we can sequence so many things and do genetically modified organisms, and all of these other things were made possible through PCR, as well as diagnostics in the clinical labs and things like that. In the early 2000s, we learned about the importance of small RNA molecules and how they can be used, pardon me, potentially therapeutically, et cetera. And then Since 2010, we've been looking at the microbiome of humans, mostly in the gastrointestinal tract, but also in other parts of the body, and trying to understand how different organisms living in different people's bodies might be impacting disease or even causing disease. So I mentioned in one of the earlier videos, things like even schizophrenia, obesity, um, things that we didn't used to think would be associated with the microorganism. We're starting to see that there are some links there, and we're gaining better understanding of how those things actually play a part in human disease. And we have CRISPR technology. We'll talk a little bit about CRISPR later in the semester when we get to the biotechnology chapter. But CRISPR is something that's a way, you can almost think of it as gene editing. So it's a way, if someone has, say, a genetic disease, like say sickle cell anemia or cystic fibrosis, that we know what the genetic mutation is, we could use CRISPR technology to go in, cut that out, and replace it with the fixed gene, the fixed information, and cure people of these diseases. And so this is another really this is going to be an a technology that's really going to change our approach to medicine and medical care, and make it much better, potentially. So how did we get to all of these great technologies and things that we can use today and things that we take for granted, like the PCR polymerase chain reaction and some of the other things? We got here by using the scientific method. And you should be familiar with this from your other science classes, so we'll go quickly through it, because again, I think hopefully this is review. But it all starts with, some kind of observations. You notice something, you make an observation, and you think, you use some information that you already have, and various observations, you put those together and you formulate a hypothesis. And a hypothesis is an explanation for what you've observed or measured. And your hypothesis needs to be something that you can test. So it has to be testable and then either supported or discredited by your experimentation and the observation and data that you make when you carry out your experiment. And so that's in a nutshell what the scientific method is. You make some kind of an observation, and from that you formulate a question or a hypothesis. You do background research to see what's already known about this. Does somebody already know this, or do I have a new question that no one's ever looked at before? So you find that, you construct your hypothesis, you test it, you design an experiment to test it, very focused and targeted to test that one thing. You look at your data, and then you either accept or reject your hypothesis, write your paper, get it published, go back, start over, add to it, whatever. It just kind of keeps going and going and going. And it's all based on deductive and inductive reasoning. So deductive reasoning is where you're using general principles to explain specific observations. So deductive reasoning, you're kind of starting with the observations and getting the hypothesis. And here, inductive reasoning, you're discovering the principles by looking at the specific cases. And we do all this inductive and deductive reasoning that kind of go together: hypothesis forming, hypothesis testing, hypothesis forming, hypothesis testing. And doing this over and over again is how we make progress in science. So deductive reasoning, again, is, for example, is knowing that opening attachments can cause damage to your computer, and so you choose not to open that attachment. Inductive reasoning is you've performed that action of opening unknown attachments, and you ultimately end up damaging your computer. So that's just one way of kind of implying inductive and deductive reasoning. So the scientific method takes time. You have to develop the ideas, you have to look at the research, you have to design experiments, you have to carry out the experiments, you have to analyze the data, You have to determine whether it fits or supports your hypothesis, and then it just kind of keeps going and going and going. Um, and if you do an experiment and it supports your hypothesis, you don't now have a theory. You still have a hypothesis that's supported, but other people have to support, other people have to test it. And other people will test it in different ways. Other people will repeat what you did, but they'll also design their own way of testing it and looking at other aspects of that particular hypothesis and that sort of thing. And after things get published and the scientific community engages with it and studies it and reviews it and continues to do testing on it, we can move forward and ultimately get to this point of a scientific theory. So scientific theories like evolution, like endosymbiosis, those kinds of things, those come from lots and lots and lots of scientists around the world testing various hypotheses that all support that particular idea. And if all of the research supports that idea, then you can have a theory where we just say, okay, we accept this. This seems to be true, a true valid explanation of this particular phenomena, whatever it is we're trying to understand. Now, it does not mean that at some point it won't be disproved. So, for example, people are still trying to test Einstein's theory of relativity, and there's been various uh, things added to it or modified by it based on quantum analysis and things like that. As we get better technology, as we get better understanding of things, sometimes what we know to be true, we have to change because we now have a better way to look at things. We now have a better way to understand them. We have to always be open in science to new ideas, to having things be restudied, and nothing, even though I know in our class, it seems like here's a list of facts. These are the things you have to know. And those things are pretty tried and true. But that being said, it's possible that 20 years from now, someone will come up with something different and we'll have to make an adaptation to our understanding when that does happen. So we have to be open. We have to understand that the science can only move as forward as the technology allows us to and as our understanding allows us to. And people will have new ideas and different ideas and ways of testing things that we just haven't even thought of yet. 20 years ago, people hadn't thought about CRISPR, um, and now it's becoming a more common thing. So, what does it take to be a good scientist and things? Well, first, ask questions, lots and lots and lots of questions, being curious. I often feel that the education system takes away our curiosity because we're all born with a lot of curiosity. Um, be curious, ask questions, wonder about things, be open-minded, be skeptical. Don't accept every single thing that you see as hard, solid fact. Look at the data, understand the data, figure out where it's coming from, what's the source of the information, how was the experiment done? Um, you have to be creative when you're designing experiments. And have, again, this comes to open mindedness, be willing to review and change your mind when evidence points to the wrong direction. So, for example, if you believed in spontaneous generation and then you have all of these facts that show you that that's not how it works, you have to be open to changing your mind. And like all humans, scientists can sometimes be a little stubborn about that, and it can be hard sometimes to change their minds. But we do. Ultimately, the data, the science will speak for itself. So early microbiology showed that microbes were everywhere, that the air, the atmosphere, the dust, lots and lots of microorganisms. You're going to do a little study in the lab about that. We do now have filtration systems very often inside buildings and things like that to reduce those numbers. But they're, so the indoors versus the outdoors, you can have different populations of microorganisms. We're breathing in. I went to a talk a few years ago, just before COVID, went to a seminar, and they said that every day you breathe in 50,000 spores of fungal spores. But we're not getting sick with those because we have systems to deal with that. But they're in the air, and we're not even aware of them. The entire earth is covered with microorganisms. They're everywhere. So some more historical figures. Already gone through a few. We have John Tyndall. He discovered that there's microbes in the dust and air and that they can have some high heat resistance. So these very resistant organisms that weren't just taken care of by boiling or increasing the heat, um, we now refer to those as endospores. And you're going to learn more about those in the laboratory and throughout our semester. But those organisms that can make endospores are things like the bacteria that belongs in the Bacillus family, as well as the Clostridium family, or genus, I should say. And these two genuses make up the bacteria or that make up endospores. And we'll learn a lot more about endospores as we go. They're much more resistant to killing and destruction than vegetative or growing bacteria. So when the organisms are growing, they're more sensitive to heat as compared to endospores. And then we have this definition, sterile, and this term sterile. We're going to learn a lot more about that throughout the semester. But sterile means completely free of all life forms. And that includes spores, particularly these endospores that are highly resistant, as well as virus particles, bacteria, everything. Now, that being said, another little thing in microbiology, the word spores can mean things that come from fungus, from yeast. Yeast produce spores, and bacteria produce these endospores, which we often just also call spores. These are two completely different things. Bacterial endospore is a survival, whereas a fungal spore is for reproduction. So this is one of those areas I mentioned earlier about terminology. This is one of those things where we have two terms that we use very frequently in biology, and they mean two very different things. If I say a spore and I'm referring to a bacterial endospore, that's something very, very different than a fungal spore. So I'm planting that seed in your mind now, but hold on to that thought as you go through the semester that the terminology can sometimes, you really need to learn it and understand. In this case, it's one word that means two different things. We'll have other times where like akaryotic and prokaryotic, where we're using two different words that we're kind of using to describe the same thing. So keep that in mind as you go through. I know it can be confusing. If you're aware of it and you say, oh yeah, that's just one of those circumstances, hopefully it'll help you gain your understanding. So continuing with our historical figures, we had the German Robert Koch, or Koch. I hear his name pronounced different ways. I say Koch because it's just easier for me. He linked specific microorganisms with specific diseases. He discovered the causative agents of various diseases. And he and Louis Pasteur, we'll talk about in a few moments, they were in scientific competition with each other. Pasteur being French and Koch being German, they were around at the same time. They were doing a lot of the similar types of work in microbiology. And apparently they didn't particularly like each other, and they would often kind of attack each other's work and attack each other even personally. But yet both of them were making big contributions to the science of microbiology that still stands up today, more than 100 years later. Along this same time, we had Dr. Oliver Wendell Holmes. He was discovering that mothers that gave birth at home had fewer infections than mothers that gave birth at the hospital. Now, think about that for a second. Nowadays, it's safer to give birth in a hospital, but back then, it was more dangerous. And the reason for that was because hospitals were disgusting. Um, that's where poor people went. They weren't kept clean. In fact, Louis Pasteur, or I'm sorry, Joseph Lister talks about when he went into the hospital that there was literally mushrooms growing out of the bedding. It was up to the families to bring bedding. Hospitals were just terrible, um, completely unrecognizable compared to how they are today. So it was safer if you were wealthy, you were able to give birth at home. The midwife would come to you, and it would just be you and the midwife and maybe a doctor, and you'd carry out your childbirth. Whereas if you were in the hospital, what would happen is there might be 10 women giving birth, sharing different midwives and doctors. They were going between patients without washing their hands, without doing anything else, and they were spreading the infections from mother to mother to mother. Um, and and women would die in childbirth, giving birth with like 20% um, you know, death rates. It was very high. Along that same time, Dr. Semmelweis, and here's a picture of him, he was noticed that women were becoming infected in the maternity ward and suggested that people should wash their hands. And when he made that suggestion that the midwives and the doctors should wash their hands between patients and also between going from the autopsy room, working with the patients that had died, back to helping a woman deliver a child, that maybe you could wash your hands between those two things. When he first made that suggestion, he actually got fired from the hospital because people knew that was just ridiculous and stupid. Why would you wash your hands? Why would I wash my hands when I'm going from one patient to the next patient when my hands are just going to get dirty again? I'm going to wash my hands before I go home at the end of the day, but there's no reason to wash them in between patients. It's just a time waster and whatever. Well, another hospital thought, well, let's just see if that works. So they hired him and they implemented hand washing. Hand washing, right? Super simple thing. We do it all the time now because we understand, but back then we didn't so much. And he reduced the deaths of women by 80%. So many women's lives were saved because of this simple act of hand washing. Um, he's my personal hero. I love Semmelweis. Simple idea, um, made a big difference, saved a lot of women. So yeah, so he's one. Another one is Joseph Lister. I mentioned him a few moments ago, talking about how disgusting the hospitals were. He acted on the hand washing, and then he took it a step further by bringing in this idea of creating an antiseptic or clean space in operating rooms. So also back then, operations were basically just hacking off legs and things like that. It wasn't like we have today, these beautiful surgical suites, everything sterile and whatnot. And so he had phenol in these misters. And so when surgeons were carrying out the surgery, there's this mist of phenol or carbolic acid is the other name for it, that would be in the air, and that was antimicrobial, and that would help reduce the infections that would occur in the patients as they were getting cut open or having their legs cut off or whatever. And it turned out that those chemicals were kind of toxic, so we don't do it that way anymore. But we built on that to now have the conditions that we have in hospitals today, and understanding the need for antiseptic techniques. Anti meaning against, septic meaning infection, so against infection. So having hand washing and those sorts of things to help with that. And we still use a lot of his aseptic techniques today. Another one is Pasteur. I mentioned before Pasteur and Robert Koch, they were around at the same time, and they were both scientists making huge contributions, but they also had a lot of conflict. And part of that was geopolitical. The French and the Germans are not known, were not friends back in the day. And so that's probably part of why they had their irritation. But Louis Pasteur invented the process of pasteurization that we still use today for milk and juice and wines and all kinds of things to reduce the number of infectious agents that are present. It doesn't sterilize, but it reduces those numbers. We'll learn more about that in a later chapter. And he also learned and discovered and gave us the germ theory of disease. So understanding that microorganisms were what was causing disease. It wasn't magic. It wasn't anything else, but actually microorganisms. Robert Koch, also kind of related in the same area, he established what we call Koch's postulates. And again, we'll learn more about those later in the semester, where it's a series of proofs to show you want to say that this particular agent is causing this particular disease. What do you have to do to prove it? And we've modified it in more recent times as we've gained an understanding of viruses and other things, because we can't always culture things or things that are specific for humans. We can't always test them in that way. So there's been some modifications of his proofs, but nonetheless, they're still valid today. And again, we'll learn more about those later in the semester. So that brings us to the end of section 6. We're almost done with this chapter, so congratulations. So you can kind of answer these or fill in the blanks. Spontaneous generation was disproven by who using the Swan-Nex flask? So you should know Pasteur. You should also remember about Francesco Redi with the meat. The polymerase chain reaction was invented by Carey Mullis. When a hypothesis is supported by a growing body of data, survives scrutiny, it's known as a theory, and aseptic techniques were developed by Lister. So, these are just some examples, but you should, go back through and answer the review questions as you go through these talks. Alrighty, I'm going to sign out and I will see you for the next talk.